IB Bio, Option B, Biotechnology, Bioinformatics, Part 5, is the first of two movies that look at B.3, biotechnology employed in the protection of the environment. Part 5, B.3A, this movie, will take a quick look at the varied metabolism of prokaryotic species, and then it will bear down on the degradation of pollutants by various microorganisms. The essential idea is biotechnology can be used in the prevention and mitigation of contamination from industrial, agricultural, and municipal wastes. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused right here. Pollutants can be metabolized by microorganisms. Various prokaryotic species have the capacity to degrade different pollutants. Various metabolic pathways break down the pollutant and release harmless products. We will define pollutants as substances released into the environment that cause harm to organisms, and this diagram illustrates a bacterial species known as Alcanovorax, seen here, metabolizing oil. In this micrograph, we can see cells of Alcanovorax borkmensis with hydrocarbons seen in black within the cell. Alcanovorax has the metabolism to degrade the oil. This is a micrograph of a species Shewanella, a deep-sea bacterium that digests PCBs or other chemical solvents. PCBs are polychlorinated biphenyls, a group of organic chemicals that with long exposure cause cancer, among other negative health effects. The metabolic pathways of microorganisms are quite varied. Microorganisms can be autotrophs. Autotrophs build carbohydrates from carbon dioxide using light or inorganic molecules as the energy source. But chemoheterotrophs derive their energy and carbon from organic molecules. And these microorganisms are the subject of this movie because chemoheterotrophs will degrade pollutants such as oil, which is an organic compound. Now you should make the connection to what we studied earlier in this option. Fermentation and methanogenesis both involve microorganisms that are chemoheterotrophs. Fermentation is one type of metabolism in which an organic molecule is degraded into a simpler substance. The term fermentation has been used in this unit to describe various metabolic reactions in which large organic molecules are broken down into simpler substances. Members of the fungal kingdom are the best known fermenters: Saccharomyces cerevisiae yeast for making beer, wine, and bread, Aspergillus niger for making citric acid, Penicillium notatum for making penicillin. You should recall these organisms and their role in fermentation from studying B.1. There are various bacterial species that also ferment organic molecules. Here are photographs of chemoheterotroph fermenters: Saccharomyces cerevisiae yeast, Aspergillus niger for making citric acid, and Penicillium notatum for making penicillin. Again, you should recall these organisms and their role in fermentation from studying B.1. Methanogenesis is also a metabolic process that starts with organic molecules and digests them, producing methane. The organic molecule noted here could be a pollutant. Here are micrographs of two methanogens. For the purposes of reminding you of various metabolic processes carried out by microorganisms, you should remember nitrification. Nitrification is carried out by autotrophs, not heterotrophs, so these microorganisms would not be those that metabolize pollutants. But redox reactions, such as this one, are employed by various microorganisms in the breakdown of molecules. And if the molecule is a pollutant, people employ the microorganism. And you should recall denitrification. Again, denitrification is carried out by autotrophs, chemoautotrophs, not heterotrophs, but redox reactions such as this one are employed by various microorganisms in the breakdown of molecules, and if the molecule is a pollutant, people employ the microorganism. Here is a list of various metabolic processes carried out by microorganisms. You should have some familiarity with all of these processes. If you are struggling to remember any one of them, return to Option B, Part 1. Microorganisms have widely varying metabolic processes, some of which degrade pollutants. And that brings us to the second IB syllabus statement of this movie. Microorganisms are used in bioremediation. And in turn, we need to define bioremediation as the use of deliberately introduced microorganisms to consume and break down environmental pollutants in order to clean up a polluted site. You should immediately see the connection between these syllabus statements and the first one of this movie, which is some pollutants are metabolized by microorganisms. So when a pollutant is released to the environment, people can employ the microorganism, bacteria, to metabolize the pollutant, thus mitigating the impact of the pollutant. Remember the essential idea. Biotechnology can be used in the prevention and mitigation of contamination from industrial, agricultural, and municipal wastes. In this diagram, bacteria take up the pollutant, whether the pollutant be oil, benzene, or methyl-mercury. They metabolize the pollutant, and then they release water, carbon dioxide, and other harmless substances. Again, here is a micrograph of Alcanovorax borkumensis that has taken up a hydrocarbon for degradation. So here is the next IB syllabus statement that takes the mitigation of pollution to the next level. Responses to pollution incidents can involve bioremediation combined with physical and chemical procedures. In the event of a spill of a pollutant, such as oil, physical methods, as seen here, can be used to mitigate the effects of the pollutant. In this case, the spread of the pollutant is being contained with shovels. Spills of a pollutant into the ocean can be contained using physical barriers, as seen here. A boom is a floating barrier used to physically contain an oil spill. Booms are used to reduce the possibility of polluting shorelines and help make recovery of the pollutant easier. In this photo, you can see the physical containment of the pollutant using floating barriers. As well, you can see the physical effort of hand scrubbing, probably along with detergents, to mitigate the effects of the pollutant in the environment. Water can be sprayed under high pressure to help break apart the pollutant. Scrubbers and detergents are also employed. As well, the water can be mixed with chemicals that break apart or chemically break down the pollutant. Chemicals can be used to reduce the impact of the pollutant. Corexit was a dispersant used to break apart the oil that was spilled in the Deepwater Horizon incident in the Gulf of Mexico in 2010. You can see the outcome of the dispersal agent here in the lower left corner. The large droplets are broken down physically into smaller droplets. This would serve to increase the surface area on which bacteria would act to further mitigate the effects of the pollutant. Corexit was used in unprecedented quantities during the 2010 Deepwater Horizon oil spill, and it became the largest use of such chemicals in the United States. Researchers continue to examine the effects and the effectiveness of Corexit. Studies so far indicate that the dispersant itself is actually toxic to marine life. In this diagram, you can see oil pouring out of an uncapped well underwater. You can see the dispersants being used to mitigate the impact of the pollutant. Corexit was used in this manner by British Petroleum in the Deepwater Horizon oil spill, when BP engineers injected Corexit at the broken wellhead, roughly 5,000 feet below the surface. Once the dispersed pollutant reaches the surface, microorganisms can be sprayed on the pollutant to metabolize the pollutant. Other chemical procedures involve the use of ozone or peroxide as oxidizing chemicals that are used to treat contaminated soils. In this diagram, you can see an oil spill here, and the pollutant is moving with groundwater flow. Bacteria are used for bioremediation. The bacteria are inserted here, serving as a biobarrier, preventing the continued flow of the pollutant underground. The injection of microbes into the polluted area allows the microbes to metabolize the pollutant. Bioremediation has been defined as a process in which microbial catalysis acts on the pollutant compounds, thereby remedying or eliminating environmental contamination. Dispersants can be released in huge quantities by planes. The pollution is emulsified into smaller droplets, which can be colonized and degraded over time by bacteria. The dispersant is a chemical procedure toward pollution mitigation, while the purposeful application of natural or genetically modified bacteria is bioremediation. The positive effect of using microorganisms to mitigate the impact of pollutants can be seen in this photograph that shows the pollutant oil in the lower portion of the photograph and the reduced evidence of the pollution in the upper portion of the image. The pollution has been mitigated by the purposeful use of microorganisms that degrade the pollutant. This is bioremediation. Here is an image of soil polluted with an organic hydrocarbon. Microorganisms, along with dispersal agents, here referred to as surfactants, are sprayed onto the oil to metabolize the pollutant. Here is another slide referencing the degradation of oil by naturally occurring bacteria. Microbes are small and they reproduce quickly, sometimes with the help of fertilizers. The degradation of the pollution results in the release of harmless products such as water and carbon dioxide. And that brings us to 3 IB Syllabus Applications. Describe the degradation of oil by Pseudomonas species. Describe the degradation of benzene by halophilic bacteria such as Marinobacter. Describe the conversion of methyl-mercury into elemental mercury by Pseudomonas. Pseudomonas species can be sprayed onto oil spills for the purpose of metabolizing the oil. A number of Pseudomonas species have been genetically modified with a plasmid that serves to produce enzymes that degrade compounds derived from crude oil. Here is a color-enhanced micrograph of Pseudomonas putida, a multiplasmid genetically engineered organism that can metabolize organic hydrocarbons. Pseudomonas putida is naturally found near or around oil. Oil-eating bacteria feed on pollutants, such as chlorinated hydrocarbons and toluene. Toluene is an aromatic hydrocarbon that is widely used in industry. Pseudomonas putida has also been used to decontaminate landfills. Pseudomonas putida is the first patented organism in the world. It demonstrates a very diverse metabolism, including the ability to degrade organic solvents such as toluene. The use of Pseudomonas putida is preferable to other Pseudomonas species because it is a safe species of bacterium, unlike Pseudomonas aeruginosa, for example, which is an opportunistic pathogen in humans. The use of Pseudomonas putida to clean up oil spills is an example of bioremediation, the use of deliberately introduced microorganisms to consume and break down environmental pollutants in order to clean up a polluted site. Now, let's look briefly at the conversion of methyl-mercury into elemental mercury by Pseudomonas. Mercury is often released to the environment by industrial processes, resulting in methyl-mercury formation in soils and sediments. Methyl-mercury is toxic because it is easily absorbed into the tissues of organisms, accumulating in concentration within those tissues that results in mercury poisoning. Now, Pseudomonas species have been genetically modified with a plasmid known as the merplasmid or the operon. The genetically modified Pseudomonas, placed on the surface of an inert medium within a column through which polluted water or soils are poured, can degrade the methylmercury, releasing elemental mercury which can be safely removed from the environment. The danger of methylmercury is that it is easily absorbed through cell membranes. Methylmercury can accumulate in the bodies of organisms, and then magnify in concentration through a food chain. Once in high concentration in organisms at the top of the food chain, it can cause severe neurological degeneration. Now, the merB operon, or plasmid, can be inserted into microorganisms, allowing for the removal of the methyl group from the methylmercury with the recovery of elemental mercury. This image displays the release of mercury by industry. This is a polluting event because various bacteria transform the mercury into methyl-mercury. Methyl-mercury is toxic because it biomagnifies through a food chain and causes severe neurological degeneration in animals. Now, bacteria with the merB plasmid produce an enzyme that safely degrades the methylmercury into elemental mercury that can be safely removed from the system. And lastly, let's look at the degradation of benzene by halophilic bacteria such as Marinobacter. Marinobacter on the left is a halophile whose natural habitat would be seawater or extremely salty conditions. Marinobacter has the metabolism to degrade benzene, the molecule seen on the right. Benzene is a hydrocarbon often found in ocean oil spills, and benzene is carcinogenic. It's known to cause cancer. Marinobacter species are well adapted to metabolize hydrocarbon compounds such as benzene in saline, often highly saline environments. Many bacteria would not survive in such environments. You can see the evidence here as films of Marinobacter cells grow well on various organic hydrocarbons. The presence of Marinobacter is seen as stained purple in this experiment. Marinobacter species will form biofilms, seen here, on hydrophobic surfaces, or in other words, on the hydrocarbons that it will metabolize. Marinobacter will form biofilms on sediments on the ocean floor, sediments polluted with hydrocarbons. Marinobacter, as a halophile, works in locations where many other bacteria would not survive. The Marinobacter bacteria in this image, the same image I just showed you earlier, are growing in films on the hydrocarbons that they are metabolizing. Marinobacter will metabolize benzene as it forms a film on the surface of the benzene. IB syllabus statement: cooperative aggregates of microorganisms can form biofilms. So let's define biofilms just for the record. It's an aggregate, film, or slime of microorganisms in which cells are embedded within a self-produced matrix of polymers adhering to each other and or to a surface. And that brings us to this IB syllabus statement that will be part of Option B, Part 6, the next movie. But as I've introduced biofilms in this movie, let me introduce the statement here. Describe the use of biofilms in trickle filter beds for sewage treatment. Polluted water, whether it be sewage or water contaminated with methylmercury or water contaminated with oil or benzene, can be trickled through a system that has bacteria in residence. The bacteria can form films in the soil or films on the roots of plants. The biofilms of bacteria can be used to clean the polluted water. Bioremediation would include the use of bacteria or plants to clean polluted water. Using plants to treat polluted water is known as phytoremediation. As the polluted water moves through the bioreactor, the trickle bed, the bacteria metabolize the pollutants, rendering the water clean. Here is an industrial bioreactor. Polluted water, particularly sewage, is pumped in at the top. The sewage water trickles through a bed composed of a medium of stones or plastic on which biofilms exist. Biofilms metabolize the pollutants, and out comes the clean water. Here is another image of a trickle bed bioreactor that uses biofilms to treat the polluted water. We have a fixed bed of rocks, gravel, ceramics, or plastics. The sewage or pollutants flow downward. Biofilms develop on the media, aerobic conditions are maintained, and clean water is the result. Biofilms can be used in trickle filter beds for sewage treatment. And that brings us to the end of IB Bio, Option B, Biotechnology, Bioinformatics, Part 5.